Hello everyone. Today we are going to discuss about the topic that is controlled drug delivery system. Most common or conventional dosage form that is used, it is solid dosage form such as tablet, capsules, which are formulated to release the active drug immediately after the administration to get or to obtain the rapid and complete systemic drug absorption. An ideal dosage regimen in the drug therapy of any disease is one which immediate attain the desired therapeutic concentration of a drug in the blood or plasma or at the site of the action, and then maintain it throughout constant for the entire duration of the treatments. So that is we can call as ideal dosage regimen. So this is possible through the administration of any drug delivery system in particular dose or dose size and at a particular frequency. So we have to identify the particular dose and particular frequency so we can get that ideal dosage regimen. So any drug delivery system we have to give, primary goal is to provide a therapeutic amount of drug to the proper site, proper absorption site or site of actions in the desired concentration or desired therapeutic concentration. So first one, we have to see what is drug delivery system. So it is a broad range of technique. So drug delivery system referred to as a technology that is utilized to present the drug to the desired body site for a drug release and the absorption. So we can call as a drug delivery system. In the control release or sustained release that we have to use the terminology. Here are some the synonyms are there that we use interchangeably in the pharma field, like they have to use sustained release, sustained action, extended release, prolonged action, prolonged release, long acting, delayed release, time based release, control release. So like that the terminology that we have to use in the pharma practice or the manufacturing part. So in that widely used the terminology that is sustained release and the control release. So what is control release? So term mention that control the release of drug from the dosage form. So control release products are designed to release the medicament or drug in a controlled manner. The first criteria, the release of drug from the dosage forms in a controlled manner at a predetermined rate, longer duration, and the site specific particular location to achieve and maintain the optimum therapeutic blood level of a drug for a specified period of time. So that is called as a control release drug delivery system. So continuous release of drug at a predictable and reproducible kinetics for the predetermined period of time throughout the course of the GI tract. So that is the control release dosage form. Second terminology, that is what is sustained release. So any dosage form or any drug release system that provide the medication over a extended time, only for the prolonged or extended time, not in a controlled manner. So that is called the sustained release dosage form. So sustained release dosage form generally don't attain the zero order type of release, means constant drug release through the time as compared to control release, and usually try to mimic the zero order release by providing drug in a slow first order based kinetic. So administration of a single dose of drug that is released over an extended period of time instead of numerous doses. So in widely we have to use the conventional dosage form, the oral solid dosage forms, but some drawback limitations are there, that is poor patient compliance, increase the chance of missing the drug because we have to use numerous number of doses, 3 times, 2 times a day, So the drug, based on their half-life, the dosing intervals are there, dosing frequencies are there, short half-life of the drug, so frequently administration of the dose, so that time poor patient compliance are there. With the numerous dose, we have to give the multiple dose, so that time the fluctuations are there in the plasma drug concentration. We are not getting the maintain the therapeutic concentration throughout the period of time, get the fluctuation. So that is the drawback or limitation of that conventional dosage form. So we have to give the sustained or controlled dosage form, the single dosage form, release over an extended period of time. So that maintain a near constant or uniform blood levels of a drug often translate into better patient compliance. We reduce the dose size and frequency, so well as enhance the clinical efficacies of the drug for its intended use. So here give the comparison between the conventional and the sustained or controlled release dosage form. So in conventional drug release, a rapid and complete release of a drug immediately after the administration, so within 1.5 hour, 2 hours, the complete drug release. In the sustained or controlled release, as compared to conventional, it go for the extended period of time, around 24 hours, 12 hours, 16 hours, 18 hours, 20 hours, 24 hours, like that. So slow or controlled release of drug over a extended period of time. In conventional, absorption is the rate limiting step for the conventional type of dosage form. So after the drug go for the disintegration dissolutions, rapidly go to the solution form at the absorption site, they go for the absorption. So here for the conventional rate limiting step, that is absorption, to control the rate process. In sustain and control, the drug release from the dosage form is the rate limiting step. So from the solid dosage form, example from the tablet or capsules, from that dosage form, drug release into that solution dissolved form in the GI fluid into the media. So that time the release mechanisms taking the role or taking the part to control or sustain that drug release part. In conventional drug release, blood level fluctuations are there, peak in valley, not constant. In sustained and controlled release, constant blood level we have to see and maintain throughout the extended period of time. So it reduces the fluctuation, so we get the better therapeutic response. We avoid unwanted side effect. So in conventional drug release, there is a risk of over medications or under medications, skip of the dose, sometimes missing the dose, or sometimes we have to take the overdose. So that times the concentration of drug getting above the maximum effective concentration go to the toxic level, so we get the side effect, unwanted effect, adverse effect, like that. So there is a chance or risk of that over medication. As compared to conventional, in sustain or control, single dose we have to take into the day, so it is reliable therapy, as the risk factor is minimum. So in conventional drug release, we have to require frequent dosings. We already see that drug dosing frequency is dependent on their half-life. If drug half-life is shorter, so we require frequent administrations, frequent numerous dose, so that compared to in sustained control release, that reduce the frequency of drug. So in conventional patient compliance we are not get, as compared to sustained and controls, with better patient compliance we get the response. More flexibility for the physicians in the adjusting dosage required in conventionals, so that time the advantageous part in that conventional, that physicians may change the dose size, dose frequency, that can flexibilities for the physician. As comparing sustains, there is less flexibilities are there, per unit cost is less as compared to sustained control, it go for the higher cost for the formulation part. So these are the comparison between the conventional and the sustained control drug release. Other terminologies are there, repeat action tablets, so alternative method of the sustained release, in which multiple dose of drugs are an alternative method of sustained release in which multiple doses are contained within the dosage form and each dose is released at a specific period, so that go for the repeated terminology. Delayed release system, in contrast, may not be sustaining since often the function of the dosage form is to maintain the drug in the dosage for some time before the release. Example, enteric coated tablet like that. So we have to delay the release up to specific period of time. After that particular condition, climate, so polymer go for the degradation, the release mechanisms taking part, and from that times the drug go for the release from the dosage form. So that is the delayed release system. Some basic history development regarding the sustain and control terminology. In early 1950 to 1970, the sustain release type of systems were developed. After that, from 1970 to 1990, control release different technologies. The work research ongoing for that control release technology or different type of control release system. In the 1990s, after that, site specific targeted drug release system go on the research on the basis of that targeted site specific particular area. The next, the difference between the two terminology that we have to use, sustain and the control release drug release system. So what is the difference are there in sustain release and control? Already we have to see the definition. In sustain release only we have to go for the prolonged or extended time period of time for the drug release. Slow drug release are there, go for maximum 12, 18 hours, 24 hours like that. Difference in control release, that also go for the prolonged or extended release, but the release rate we can predetermine. We can control the release rate from the dosage form, so we have to maintain the level of drug release from the dosage form and maintain the drug concentration in the plasma. In sustained release, do not attain the zero order base release kinetics. In control release, we have to nearer to zero order kinetic process we have to get. The release is based on the concentration in the sustained release. In control release, they are not dependent on the drug release, that is based on the drug concentration. Sustained release as compared to control release is not a site specific targeted, but we have to go for control release, we have to go for the site specific type of. So these are some difference between the sustained and the control release type of drug release system. So here in the figure we have to see the plasma drug concentration versus time. We have to plot the graph. So in that we have to see the conventional type of things, sustained release and the control release. So for the conventional type of drug release system, we have to go for multiple dosing. In that case, what we have to see it in the multiple dosing, in some case the fluctuations are there, some case we have to go for the below the minimum effective concentration, it means subtherapeutic level. That concentrations are not giving any pharmacological actions, and some multiple type of dosing that go for the above the maximum safe concentration or maximum effective concentration, it go for the toxic level, so that also giving unwanted effect. So that fluctuations we have to face in the conventional dosage form by the multiple dosing. As compared to that, we have to go for the sustained release, so we have to get only the prolonged or extended period of time the drug release are there. And the third one, that is our control release drug release system. So in that we have to see that the concentration is maintained in between the therapeutic level, so that is zero order control release base. So drug or the therapeutic concentration in the plasma is maintained in between that minimum and maximum effective concentration range, and we get the effective and better therapeutic action. Another schematic diagram figure of that sustained or extended control and compare with the immediate or conventional release. So in that case, the plasma concentration versus time, first we have to see that figure. So in that The first, the black line, that is the immediate release. The example for that, so within one and a half an hour we get the maximum drug release. And then we have to see the second, the orange line, that represent the delayed release. So we have to see that in the figure. So up to 0 to 2 hours there is no drug concentration found in the plasma. After 2 hours drug going for releasing from the dosage form. And the blue line, that is for the extended release. Similar diagram for the percentage drug dissolves or percentage drug release versus time. Similar points are there for that, for the immediate, extended and the delayed release. One more examples for that, for all the terminologies that we have to see. So here in the figure they mention the hypothetical in vitro drug release characteristic from various oral dosage forms. In the first diagram, the image, that is for the conventional tablet, conventional dosage forms. That type of things are there in that. The B, that is for the delayed release. So up to 2 hours, 3 hours, there is no drug release from the dosage form. After that, the curve we have to see for the drug release part. The C image represent the repeated actions. So here we have to see the first, the curve increasing. After that, repeated actions, further the graph going for increasing the release of drug in the plasma. The D represent the prolonged release. D and E are the similar terminologies, are there synonyms. We have to use prolonged release, extended release or sustained release. They go for the release of drug up to 24 hours or prolonged extended period of time. And our fifth one, that is the linear, we have to see the graph with the time, the concentration maintained throughout the period of time for the extended or prolonged period of time, that is control release. So the F, the image represent for the control release type of dosage form or drug release system. So one more image are there for that. So drug level in the blood, with here the examples we give. The first one, that is for the traditional or conventional or immediate release dosage form. So in that time the multiple dosing we are given. So in that we have to see that the therapeutic range, minimum effective and maximum effective concentration. So after the multiple dosing, every dosing, first, second, third, the dosings we are given, so that go for, sometimes fluctuation go for higher than the maximum effective, so it go for the toxic level. Sometimes go for the subtherapeutic level, below the minimum effective concentration. So fluctuations we have to face with the traditional or conventional immediate release drug release system. And the B diagram, that is represent the control release drug release system. So we have to see that they maintain the concentrations within the therapeutic range throughout the specific period of time for the prolonged time. Now we have to see the advantage and disadvantages of the controlled drug release system. So what are the advantages are there? So it improves the patient compliance. Already we have to say that we have to reduce the frequency as compared to conventional. Number of dose we have to reduce, so that we get the patient convenience and compliance due to the less frequent of the drug administration. Second, the reduction in the fluctuations. Already we have to see in conventionals, multiple dosing we have to see go for the above and below the concentration range, therapeutic range. So here in controlled release, so fluctuation we go for the reduce, we get the steady state level, and therefore we get the better control of the treatment disease conditions, we get the better therapeutic response. Increase the safety margins of the high potent drug, because it maintain the concentration within the range. Throughout the concentration we have to maintain in the plasma. So for the highly potent drugs, safety and efficacy for the margins are increased. We have to go for maximum utilization of drug, enabling the reductions in total amount of dose. So reduce the dose frequency by using the optimum utilization of that drug. Reduction in healthcare cost through the improve the therapy. We have to reduce the dose dosing frequency for the shorter period of time, so we can improve the therapy by the reducing the cost, reduction in the GI irritation or local or systemic side effect or adverse effect that we have to reduce by reducing the dosing frequency and dose size, as well as we have to go for the site specific targeted, so that also helpful. And it enhances some case in it enhances the viability of drug by applying the control drug release system, and we have to achieve the targeted site specific drug release. The disadvantages are there for the control drug release system, that prompt the termination of therapy is not possible if you get the single dose for the 24 hours as compared to conventionals. The termination part, it is very complicated. Less flexibility for the physician to change the dose size, dose frequency, dosage regimen, changes in the dose. It is less flexibility as compared to conventional dose dumping. Maybe possibilities are there. Problems due to variation, so many factors, the economic factors as well as some factors, the biological and the physicochemical factor of drug, that also alter the or create the problem in that control release formulation. All the drugs are not suitable for the control release. Specific criteria required for the drug to go for the control release type of dosage form. In some cases, stability problems also arise in that control release part. So these are the advantages and disadvantages of the control release drug delivery system. What are the rational goal or objectives for the development of the sustained release or control release drug delivery system? There is the few points are there, few rational points are there. That's why we have to go for sustained or control release type of drug delivery system. The first one, to extend the duration of action of the drug. We have to control or go for the slow release from the dosage form. We have to get the extended or prolonged period of time. To minimize the fluctuation of drug in the blood or plasma level, so you have to improve the drug utilization, optimum use of that drug, and to reduce the frequency of dosing and providing the uniform drug use system. We have to maintain the concentration. We have to go for targeting part. We have to get optimum utilization of drug, reduce the frequency. So these are all the rationals, strong rationals for that. So we have to apply for the sustained or control release type of drug delivery system. Next, the most important parameters for the selection of drug candidate. The ideal drug candidate that is suitable for the control release formulation or any sustained release formulation. So here are the some points are there. The properties of drug that is not suitable for the or ideal candidate for the drugs that having that property, so it is not suitable for the control release. Very short or very long half-life, that is not suitable. They are having a significant first pass metabolism, poor absorption throughout the GI tract, not suitable. Low solubility, low aqueous solubility, so that also not convenient to go for the control release formulation. Large number of dose, more than 500 mg or 1 gram from that. So larger the dose we have to incorporate in the dosage from that, also not suitable. Narrow therapeutic window. So these are some points that we have to go for the selection of drug candidate for the control release formulation. So for the any type of drug release system, novel or control type of drug release system, we have to prepare. So we have to require the polymers. That is the most important part for the drug release system, or getting most important role in that for the control or prolong the release of the drug. So that polymer have some ideal characteristics are there. They are chemically inert, biocompatible, the mechanically strong, comfortable for the patients, comfortable for the manufacturing part, also for the production, capable of achieving high drug loading capacities are there, safe from accidental release. We have to go for control that times, easy to fabricate, easy to sterile if needed. That type of parental drug release systems are there. So there are some ideal characteristic criteria required for the polymers that use in the sustained or control drug release system. The factors that we have to consider during the preparation or formulation of any controlled drug delivery system. So various factors are affecting the sustained or controlled drug delivery system. The first, that is route of drug delivery. So that also alter the control release mechanism, drug release mechanisms. We change the route from oral to parenteral or extra widely. We have to use oral drug administrations. Type of drug delivery systems, that also changes are there based on route. You have to go for transdermal, you have to go for buccal drug delivery system, gastroretentive, colon, ocular drug delivery system, osmotic drug. So that type of drug delivery system also we have to consider during the control release formulations. Based on their drug delivery system, formulations are altered. Disease being treated, patient, the length of therapy, the acute or chronic, the period of time that treatment's gone, and the property of drug, the drug physicochemical profile. So these are the factors that we have to consider before we have to go for the formulation of any controlled drug delivery system. So that the biological factors are there, the absorption, distribution, metabolism, the half-life, duration of actions like that, margin of safety, circadian rhythms. So for the absorption, so they have to absorb throughout the GI tract. That is criteria required. The drug release in that stomach or in that intestine by the passive diffusion, they go for the absorption. So that are criteria is required. Distributions, API with large volume of distribution is not suitable for that candidate, is for the control release. API should be metabolized with intermediate speed, not highly metabolism, high first pass metabolism or metabolism processor. That is also not suitable, that affecting to the, if go for the rapid metabolize in the livers, so that is not an ideal or good candidate for the oral controlled drug release system. So that gives with the other route, other than oral route, we have to go for the parenteral like that, because rapid metabolisms are there, they not go for the absorption, they not give the therapeutic response, so that is not ideal. The half-life, the biological or eliminate half-life is in between 2 to 8, so it is ideal, excellent, it is ideal candidate for the control release. If less than 2 or more than 10 to 12, so it gives the sustained activity or action, so it is a sustained release type of dosage form, so that not go for further the sustained or control type of release for the longer half-life. Margin of safety, so high enough so dose dumping does not cause any serious side effect. If go for the dose dumping, we have to give the large number of dose in sustained release, and that go for rapid, sometimes rapid release type of things happen, so that giving the serious side effects like that. So we require some margin of safety also in that case. Plasma concentration response relationship, say linear relationship, is better candidate. Some physicochemical factors, and that dose size. So ideally, if we can go for the sustained or control release type of dosage form, so we require less than 500 mg is ideal dosage form for ideal dose size for the candidate which go for the sustained or control release, but maximum 1 gram. Molecular weight, so less than 600 Daltons are suitable, because if molecular size or weight increasing, so that is affecting the difficulties into the absorption part, because for the absorption we have to go for by the passive diffusion, and for the passive diffusions they have to cross the cell membrane, and that time ideal criteria require, that is the particle size or the drug molecule size is less than 500 or 600 Dalton, easily cross the cell membrane by the passive diffusion. So larger molecules are not cross the cell membrane, not go for the absorption by passive diffusion, they go for the other mechanism like endocytosis or carrier mediated transport like that. But the less than 600 are ideal candidate for the sustained control release type of. Then the solubility, the aqueous solubility we have to require, because for the absorption, the drug must be in a solution form at the absorption site. So the solution form that we would require the solubility of drug to the particular medium, GI fluid or aqueous fluid. So drug having a sufficient aqueous solubility. So drug having both the natures we would require for the absorption by passive diffusion, hydrophilic and hydrophobic. So for hydrophilic water soluble drug we would require for the solubility, soluble or dissolves into that particular media and get the in a solution form. Then after they go for the absorption or permeable part by the passive diffusion through the cell membrane. So that time we would require the lipid nature, because cell membrane contain biophospholipid. So lipid drug cross the cell membrane and reach to the blood for the complete the absorption process. So good aqueous solubility with the pH independent solubility, it serves as a good candidate, because in throughout the GI tract pH may vary from 1.2 to around 6 throughout the GI tract up to 7. So in that case, if pH based dependent solubilities are there, so changing the pH from acidic to alkaline or basic, so solubility also altering. So that's why pH independent solubility candidate is ideal for the control release type of the form. Drug with pH dependent aqueous solubilities are there. So drug soluble in non-aqueous solvents, some suitable candidate for parenteral type of control release system. Poor water solubilities are there. BCS class 2 drugs are there. Low solubility, low aqueous solubilities are there. So that times we have to improve the solubility by solubilization technique. Then the ionization. The pK value, dissolution constant or ionization constant that we have to call, so that is based on the pH. So for the absorption of drug through the passive diffusions, the criteria requires that unionized drug that easily cross the cell membrane. So for the absorption of any drug, so that require at the absorption site it a unionized form, so they cross the cell membrane and they reach to the blood or plasma. So the drug remain in an unionized form at the absorption site is a good candidate. But if particular pH in the GI tract drug go for the ionized state, so it convert into the ions, so they are not cross the cell membrane by passive diffusion, so that time the absorption viability problems arise, so that is poor candidate for the drug absorption. So that also we have to see in the sustained release control is part. Partition coefficient, already we have to see that aqueous solubility is required, similar like lipophilic city or lipophilic nature also required. So that we have to find out with the partition coefficient, the log P value. So that require more than 1 or 2. So if lipid solubilities are there, they are easily permeable or cross the cell membrane, they reach to the blood. Like that protein binding, higher the protein binding, so itself it gives the sustained hypothesis, because bounded drugs not go for the metabolism action, they not go the any pharmacological action. So less protein binded is ideal suitable for the sustained control. Stability, if drug is unstable throughout the GI tract, in some case it not suitable in stomach, they go for the degradation. So that is poor candidate, because that go for the degradation, that alter the viability problems, that not give the any therapy response. In some case it is not suitable in intestinal, the alkaline medium, that also the case. So the drug is stable throughout in GI tract, that is ideal candidate for that part. So here we have to see the aqueous solubility for the drug that we have to require, that is more than 0.1 mg per ml. So that is suitable, that we have to go for the sustained control release type of dosage form. The partition coefficient, we have to require the more than 2, that is ideal, the log P value. Drug stability in vivo highly enough so drug remains stable throughout the drug release for the period of time. Protein binding, so drug with high protein binding will not require for the release modifications. Drug pK ionizations at the physiological pH, so ideal for pK for acidic nature of drugs, so in between 3 to 7.5 that is suitable. For pK base, the less than 11 that is ideal, there is no problems for that, the drugs for the sustained control type of drug system. Mechanism and site of absorption, so drug absorbed by the carrier mediated transport, those absorbed through the absorption window, the poor candidates are there. So absorption should not be active type and absorption window should not be narrow, so that is suitable for the sustained control. Molecular size, already we have to see size and diffusivity less than 500 600 are easily diffused to the matrix and the cell membrane, so that dose size greater than 300 mg, or sorry, less than 300 or less than 500 mg also suitable for that sustained control parts. So the next, that is general structure of control release. So in that the core parts are there that containing the drug and the additives or excipient, that is filler, lubricants like that, general tablet ingredients that we have to use in core part. After that the outer layer, that is coating materials, the coat that is made up of the polymers, also drug we can add and the lubricant, but major part that is polymer. The next one, that is the mechanism aspect of oral drug delivery system. So they classify into the 4 types: dissolution based control release, diffusion based control release, erosion control release, and the combination of both dissolution and diffusion. Further, the dissolution control and diffusion control further classify the matrix and the reservoir. We have to see one by one. The first one, that is dissolution control release. So solid substance that is solubilized in the given solvent, so that is process called the dissolution. Or solid materials that dissolve or soluble into the given media or the given solvent. Otherwise you can call mass transfer from solid to liquid, that is called the dissolution process. So that is the rate determining step for that, the diffusion from solid to liquid. Various theories are there to explain the dissolution process. In that, widely we apply the diffusion layer theory, stagnant layer theory. Other than that, surface renewal theory, limited solvation theory. So there is the 3 theories are there which explain the dissolution process. We have to express the dissolution process or solubility process by the Noyes-Whitney equation. Dissolution control is further classified into encapsulation based and the matrix type of dissolution release. The first one, the encapsulation. So in that core part the soluble drugs are there, and the outer layer coat materials, that is slowly dissolving or erodible coating materials that we have to use, so that is the encapsulation. In the center part drug is there and we have to encapsulate or cover, laminate with that polymer coating. So drug that coat with the slowly dissolving and the permeable polymeric materials. So drug release controlled by adjusting rate limiting step, that is thickness and the permeability of that coat. The coating polymer material that we have to use, their thickness and the permeability parts. So membrane coated particles, the compress into the tablet or place in the capsule, that is the example. Coating we have to done by the one of the most important widely used technique, that is microencapsulation method, that we have to apply for the coating of the tablet for the encapsulation part. So here the process are there, micro encapsulation, various techniques are there. In that most widely used the co-adsorption or phase separation technique for water soluble polymers. Other than that, so many techniques that we have to use: salting out, hot melt, solvent evaporation, precipitation, interfacial polymerization are there for the both water soluble, solvent soluble polymer. For second type, that is matrix type. So in that the drug and the polymers are homogeneously distributed throughout the matrix. So in that the soluble drugs are in the center part with that slowly dissolving or erodible matrix. So it is also called as a monolithic type of system or monolithic dissolution control system. Here the controlling the dissolution rate by the mechanism we can call the 3: altering the porosity of the tablet. So this is the rate limiting step for that. From the matrix type of dissolution control system, we have to go for altering the porosity, decreasing their wettability part, or dissolving at the slower rate. We can reduce that three parts, and then we can control the dissolution based drug release. Rate controlling medium or matrix that we have to use in that case, examples are beeswax, carnauba, like that, the castor oil, like that we have to use for preparing the matrix type of system. For the dissolution control based drug release mechanisms, we have to explain by the Hixson-Crowell cube root law. So here the theory for that Hixson-Crowell cube root law, for the drug in a powder form, that drug particles consisting of uniform spherical size particles, and the rate of dissolution is expressed based on the cube root of the weight. So that is Hixson-Crowell cube root law, that express the dissolution into the cube root of the weight of the particles. So the radius of the particle is not assumed the constant. With the time, the radius of the particle is decreasing. So drug powder consisting of uniform size spherical particles, that we have to assume that particle size of the drug, it is in spherical and uniform. Dissolution is effective from the surface of the particle. That is common for the dissolution part, the dissolution theory, that the drug is dissolved or soluble from the surface area. So surface area of the powder, we can calculate the number of particles multiply into the surface area of each particle. So here the surface area of the particle S is equal to N into 4 pi r square. So N is represent the number of particles, and surface area, the equation, that is 4 pi r square. So now during the dissolution process, with the time, the radius of the particles is reduced. Change in the diameter and the size of the particles from the surface, drug go for the dissolution, so the particle size go for the reducing. So change in the volume of that powder, so dV is equal to N into 4 pi r square dr. So during the dissolution, mass of the particle also go for the decreasing, similar like radius. So change in the mass is equal to, the equation becomes the density of powder into change in the volume. So that we have to apply for that part. So then we have to go for the rearrangement substitution for that, and go for the integration, and then we have to get the equations for that, and lastly we have to find out the equation for the Hixson-Crowell cube root law. The particle size decrease and change in their surface area that accompanying the dissolution. So Hixson-Crowell cube root law for the dissolution is used by the m01 by 3 minus m1 by 3 is equal to kd. So the cube root law of the mass. So m0 is the original mass, initial mass of the drug. M represent the mass of the drug remaining to the dissolves at the time t, and k is the dissolution rate constant. Second mechanism, that is diffusion control release. So it is a major process for the absorption, no energy required for the diffusion parts. Drug molecule diffuse from the region of higher concentration to lower concentration until the equilibrium is attained, that is based on their concentration gradient. So that is directly proportional to the concentration gradient across the membrane. So in that also the reservoir and encapsulation of matrix type systems are there. So reservoir similar like the encapsulation, in the core part the drug is there that hold the drugs in the reservoir. Outer layer, that is made up of polymer, that is insoluble membrane, and drug is diffused from that core material to the bulk or outside. So here the rate limiting or rate controlling step, that is polymeric contain the concentration of polymer in the coating material, the thickness of that coating material or coating layer, and the hardness of that microcapsule. So that also affect into the release part. So it is also called the laminated matrix device, the hollow systems that is containing inner core materials surrounded in water insoluble materials water insoluble rigid material. That's why it's called the laminated matrix device. The polymer can be applied by coating or the microencapsulation. Similar already we have to see in that dissolution based control. The coating we can done by the any microencapsulation technique. So the rate controlling mechanisms partitioning into the membrane with subsequent release into surrounding fluid by the diffusion. The common examples they are given for the polymers that use in diffusion control. The reservoir type, the HPC, ethyl cellulose, polyvinyl acetate, like that. In matrix diffusion system, the swellable gum matrix that we have to use sent with the drug, swallow and glossy hydrogel approach, from that which drug has completely diffused. So here the two approaches are there, rigid matrix or swellable matrix. Here the rate controlling step, that is diffusion of dissolved drugs in the matrix. So rigid matrix diffusions in that materials that we have to use, that is insoluble plastic such as PVP, fatty acid, that is rigid material. In swellable matrix diffusions, they go for forming the gel. When they absorb the water, they increase or go for the swellable, so that create the gel type structures, and from that they go for the releasing or diffusing the drug. So also called as a glassy hydrogel. It is popular for sustaining the release of highly water soluble drugs. Materials that is used, that is hydrophilic gums are there, natural gum, guar gum, tragacanth, xanthan gum that we have to use, and HPMC, CMC. Like that, the examples for the material that is used is a swellable type of matrix diffusion. Previously, for the dissolution control drug release, we have to apply the Hixson-Crowell cubic law mechanism. For the diffusion based mechanism control release, so that we have to find out with the Higuchi equations. So here the drug release from the homogeneous polymer matrix. So here the equations for the Higuchi, that is Q is equal to D into bracket 2A minus CS into CS into T, there 1 by 2 under root of that equation. So that is the Higuchi equations. So further go for the modification in the Higuchi equations, we have to add the granular matrix. So the equation go for the modifications. In that equations we have to add the porosity and the tortuosity, that we have to counting. So here Q is the amount of drug that released per unit surface area at the time. D is for the diffusion coefficient of the drug in the release medium. Epsilon is for the porosity of matrix. CS represent the steady state or the solubility of the drug in the release medium. T for the tortuosity, and A is represent the concentration of drug present in the matrix per unit volume. So that is the equations for the Higuchi model. Tortuosity, that we have to apply that. What is that tortuosity? To account for an increase in the path length of the drug diffusions. So for the diffusion pathway, they go for the branching and bending of the pores as compared to short straight pores. So that we have to consider as a tortuosity. The third mechanisms for this control release, that is erosion based control release. So these materials that degrade the within the body as a result of any natural biological process, that is chemical based or enzyme based degradations are there. So they are further classified that bulk erosions and the surface erosion. So bulk throughout the homogeneously the matrix are there. So the polymer degrade in fairly uniformly throughout the matrix. So that is bulk erosion, and the degradation occurs only at the surface of the polymers. So that is called the surface erosion. The resulting in release rate, that is proportional to the surface area of the drug release system. So here in the diagram that represent the bulk eroding and the surface eroding biodegradable polymer that we have to use go for the erosion. The last one, that is the combination of both dissolution and diffusion based control release system. So both the approach that you have to apply, the outer layer that is made up of water soluble and water insoluble, both the polymer, the hydrophilic and hydrophobic polymeric materials are there. So that we have to prepare the coating membrane. So here the center part, core part, that is contain the drug. Outer layer, it is made up of semi permeable membrane. So in that case, the water soluble hydrophilic polymers, when they contact with the liquid, they go for the soluble form, they create the pores, and the liquid penetrate into that core part, they dissolve the drug into that particular core area, center area. From that, the dissolved drug go for the diffusions, and that go out from that the dosage form. So here the drug enclosed in partially soluble membrane. So center part, it is closed with the semi-permeable partially soluble membrane, with hydrophilic, hydrophobic, both the polymers are there. So pores are created due to the dissolution of the part of the membrane. That hydrophilic polymers go for the dissolution, they create the space or pores from that place. So it permit the entry, they gave the entry of that aqueous medium into the core part. The aqueous medium enter into the core part, they contact with the drugs, they go for the dissolution process inside that core part, and they dissolve the drug, and dissolved drug diffuse out from that system. So that is the dissolution diffusion combination therapy. So the coating is used as a matrix of ethyl cellulose with methyl cellulose. So ethyl cellulose is a water insoluble hydrophobic polymer, and methyl cellulose or HPMC, that is used for the water soluble or hydrophilic parts. So the methyl cellulose or HPMC dissolves in the water, they create the pores. Ethyl cellulose is resid insoluble, so it stay like that. Liquid penetrate into that pores part, they dissolves the drug, and dissolved drug diffuse out from that membrane. So here we have to see the basics of sustained release control is their introduction, the ideal characteristic of drug which is suitable for the sustained release control is, difference between conventional and sustained control, comparison between sustained and control, advantages, disadvantages of the sustained release control release, which are the factors that we have to consider for the forming of any sustained control release dosage form, and the mechanism of control release system.